So it is my great honor to introduce today's speakers, Professors Donald and Ada Olins. As you remember, several seminars ago, we hosted here the talk of Caroline Luger, who published the first crystal structure of the nucleosome. And so today, we have here Don and Ada, who were the first to discover that the nucleosomes even exist. Don and Ada probably will not mention about the nucleosome discovery story today, as they will be talking about their recent research. So let me briefly update you on the history. So nowadays, it's quite trivial for us that the DNA forms beads on the string structures as the elementary unit of genome compaction in the cell nucleus. But imagine that in 1970s, scientists didn't know that. In fact, when another chromatin pioneer, Chris Woodcock, suggested that it's possible to have such structures, then the reviewers' comments were that if that were to be true, that would have required rewriting all textbooks. Luckily, when Don and Ada submitted their paper reporting the first microscopy images of nucleosomes, they somehow managed to convince peer reviewers that these beads on the string structures indeed represent the real state of our genome. And their paper was published in Science in January 1974. At that time, they called the structure new body, but later it became known as the nucleosome. So at least the first letter survived. A few months later, a paper reporting the existence of nucleosomes was also published by another pioneer of this field, Roger Kornberg. And later, indeed, all textbooks had to be rewritten to take into account the nucleosomal DNA structure. So the discovery of the nucleosome happened 46 years ago. At that time, Don and Ada were both working at the University of Tennessee, Oak Ridge. Before that, they had to change several places to be able to stay together. Ada had initially started her studies in Harvard, but decided to switch to New York to be together with Don, where he was getting PhD from the Rockefeller University. And so Ada finally obtained her PhD from the University of New York. They made an amazing pair of wife and husband, as well as the closest scientific collaborators, and worked together. And scientists who were lucky to collaborate with them know that Don and Ada even signed their emails together. And their email signature is DNA, which stands for Don and Ada. So after getting PhDs in New York, they both did postdocs at the Dartmouth Medical School in New Hampshire, and then obtained permanent positions in the University of Tennessee, Oak Ridge. Don and Ada worked at the Oak Ridge for 30 years, then for several years at the Bowdoin College, before moving to the University of New England, where they are still affiliated as research scientists. So at this very senior stage of career, many scientists would just enjoy retirement, but that's not Don and Ada. In fact, all results that they will be presenting today were obtained after their retirement, and their productivity during this period is amazing. What is most striking is that they not just supervised someone, but actually they did the experiments, the wet lab experiments by their own hands, and traveled a lot as visiting scientists. It was during one of such experimental projects that they were performing as visiting professors in the German Cancer Research Center in Heidelberg, where we were introduced to each other, and we published 3 papers since then. And one of these papers actually was published this week. So finally, I would like to say that this couple has been an inspiration for many scientists, and an example how dedication to science and dedication to family can go hand by hand. And definitely, they're an inspiration to me. So, welcome to the presentation of Don and Ada.
Thank you so much, Vladimir, for that very generous introduction. And thank you all for inviting us to participate in this very impressive seminar series. We've enjoyed listening to the earlier speakers, and we think it's actually one of the nice features that we can watch and listen to scientific talks while we're isolated in our homes. In fact, we're in our home right now, and because we are, I want to say that our rules at home are that Don and I can interrupt each other. So please don't be upset if we correct or interrupt each other during the talk. I'm not going to present much historical information. I just wanted to show this one electron micrograph of the beads on the string, your fragile nucleosomes. And at this point, recall the excitement when Chris Woodcock and we had abstracts back to back at a cell biology meeting in Florida, and shared our new data on the chromatin structure. Sadly, Chris passed away a few months ago, and I wanted to pay tribute to his contribution to chromatin. So let me start with a movie that many of you have seen. This is a neutrophil chasing a bacterium, and it is, let me get it started. All these round particles that are red blood cells which have no nuclei. The neutrophil or granulocyte, which is chasing a bacterium and trying to protect from a bacterial infection, is a very flexible molecule. It has a nucleus, a very interesting nucleus, and that's really why I'm showing you this movie. Since I can't hear all of you cheering when this neutrophil caught his bacteria, I'm going to go on to the next slide. And this is a thin section electron micrograph. I'm going to warn you, we have a lot of micrographs, and I hope you enjoy looking at structure. I'm going to compare confocal microscopy, STED microscopy, electron microscopy, cryomicroscopy, and really emphasize how being able to see structures of biological systems has been so important to our understanding of chromatin. So in this micrograph, there are two granulocytes that are in the lumen of a blood vessel, and the you can see that there are, here's a red cell, here are two granulocytes, and the very characteristic nuclei of granulocytes is that there's a lot of condensed chromatin and that the nucleus is lobular. It's not a spheroid nucleus, it's a very many lobules of one nucleus. And what you can see here that really demonstrates the importance of the lobulated structure of the nucleus is granulocytes squeezing through the endothelium of the blood vessel. And these spaces sometimes are as small as 1 micron. And so the cell has to really change its shape dramatically. And the nucleus also changes, and that affects both transcription and structural aspects of the nucleus. So this dramatizes how well, how malleable, how plastic the granulocytic cells are. So we decided to study a tissue culture line that could be differentiated with simple chemicals, retinoic acid and 4-ball ester. And so this cell line is called HL60. It's a S4. It's a derivative of HL60 cells. This is taken from a human leukemia patient many, many years ago. And these cells grow in culture very vigorously. They have a large nucleus. They have prominent nucleoli. When you treat them with retinoic acid, they will become granulocytic cells, and the nuclei will become lobulated. if you treat them with 4-ball ester, they will become macrophage attached to the vessel that they're growing in, and the nuclear shape remains round. So these are very dramatic changes that we can, within 4 days, stimulate just by these 2 simple chemicals. We've done a lot of work with these cells, and they're really a staple to our laboratory. Let me now go to a little bit of fundamental aspects of, I guess
It's okay, forget it.
let me just go over the basics of what the surface of the nucleus looks like. The surface is connected to the endoplasmic reticulum. So this way, going to the top of this diagram is the cytoplasm. There's an outer nuclear membrane, ONM. There are pores in the nuclear envelope, just shown with a line here. Of course, these are one beautiful structures in the wall of the nucleus. And then there's an inner nuclear membrane. There are, I just looked up over 100 proteins that are part of the nuclear envelope membrane. So I'm just showing here a few Lem domain proteins that are in the inner nuclear membrane. And I especially want to emphasize Lamin B receptor, LBR. We'll be talking about this fantastic protein quite a bit in the next few minutes. Then underneath the nuclear envelope is a layer, a lamina, made up of various lamin proteins. These are similar to intermediate filament proteins. And then there is a layer of chromatin. Now, the thing to notice is that there are a lot of dotted lines here. These are all connections between the proteins, between the lamina, between the BAF protein, which connects many of these proteins to chromatin. And I'm going to, if you notice, the chromatin here has a thin layer at its surface, which is labeled epichromatin. And epichromatin is something we've been concentrating on quite a bit. And this is a very thin surface of chromatin, the layer that's closest to the envelope. And it is different in its structure, and that's one of the things I want to emphasize this morning. The next thing I want to just show in a cartoon style is the connection between the nucleus and the cytoplasm. There are SUN proteins. This shows the SUN1 dimer, and it connects from the inner nuclear membrane via Nesprin proteins to the cytoplasm. In one direction, the protein Nesprin 3 connects to plectin, which connects to intermediate filaments. In the case of the HL60S4 cells, these are vimentin. On the other side, there are Nesprins which connect to actin filaments. So this is a very complicated and well-organized system that holds the nucleus in place, so to speak, and allows flexibility or makes the cell more or less stiff. So this link system, linker of nucleoskeleton and cytoskeleton, is a very important part of how flexible, how deformable these cells actually are. So let me show you some transcriptome data. When we go from cells that are just growing to our retinoic acid treated cells, those are the blue lines here. And on the other hand, comparing the TPA, the 4-ball ester treated cells. And I don't want to go through all the nuclear envelope proteins, but I want to emphasize several. Lamin A cells are treated with retinoic acid is very much decreased. And lamin A is the intermediate filament, the nuclear lamina protein that makes the nucleus very stiff. So this is in line with the property we see of the more flexible nucleus. In TPA treated cells, lamin A goes up. I also want to point out that Lamin B receptor, which seems to be involved in nuclear envelope membrane synthesis and also in adding flexibility to the nucleus, goes way up with retinoic acid treatment. And here, SUN2 also goes up. SUN1 goes down. And more transcriptome data, and I'm just going to show two aspects of the cytoskeletal proteins. One is plectin, which goes down when you treat with retinoic acid, and the other is vimentin. So the changes in the proteins when you treat with retinoic acid and form granulocytic cells is very much in line with presenting a more flexible, more deformable cell and nucleus. Let's just go quickly over some of the things we've seen. That lamin AC is very much decreased in granulocytic or retinoic acid treated cells. Lamin B receptor, which I'll talk about a little more, is increased, and the entire link complex is pretty much weakened, decreased. We've also had the pleasure just a few months ago, before COVID hit, to work in Jochen Guck's lab in Erlangen in Germany. And they have a fantastic machine that measures deformability. They can scan thousands of cells, image the cells, and see how much they deform by migrating through a very narrow capillary. So those measurements also showed that retinoic acid treated cells, granulocytic form of HL60, are much more deformable. So what do we know about Lamin B receptor? It's a beautiful protein that stains the outer surface of interphase cells. When the cell goes into mitosis, it goes into the endoplasmic reticulum. It doesn't stay on the chromosomes. When we look at electron micrographs, and these four panels are electron micrographs, the top left is a cell preparation that has not been treated with retinoic acid. So you see the nuclei are fairly ovoid. They're somewhat irregular, but they don't have any lobules. When you treat with retinoic acid, and the other three panels show that, at lower magnification, you can see that the nuclei become lobulated. Here at a little bit higher magnification, you start seeing some kind of structure between the lobules. And let me just jump right to the bottom right panel, which shows the structure of these what looks like strings in the left panel. These are really sheets. We call them envelope limited chromatin sheets. ELCS, ELCS. And the structure of the ELCS is as follows. We're going here in the direction of this arrow, outer nuclear membrane, inner nuclear membrane, and then a layer of chromatin, which looks very organized, and then another inner nuclear membrane and outer nuclear membrane. So try to imagine this is a section cut through a nucleus, and we're cutting perpendicular to this sheet so that we can see the membranes clearly, and we can see the layer of chromatin quite clearly. I'm going to talk a little more about that in a minute, but I wanted to show you that in the confocal microscope, here the panel on the left, now this is not in the electron microscope, but we can see the same structures. In cyan, you can see the DNA, and in red, Lamin B2, which is a lamina intermediate filament, which is also a part of the ELK system. If we knock down Lamin B receptor with a short hairpin methodology, we find that the differentiation with retinoic acid is changed quite a bit. So these are, again, electron micrographs, thin section. The upper left corner is a growing cell, untreated with retinoic acid, an ovoid nucleus, a nice nucleolus, and a layer of chromatin right on the surface that is more pronounced than the rest of the chromatin. When you treat with retinoic acid, you get lobulation, just as we saw in the previous slide. You still have nucleoli, and you see the ELKs quite clearly. In the knockdown cell, where there's no Lamin B receptor synthesized, the cells grow, surprisingly, they grow very nicely. And when you treat with retinoic acid, they continue to grow, but there's no lobulation and there are no ELKs formed. So Lamin B receptor is obviously very important for lobulation and ELKs formation. Let's look at Lamin B receptor a little more closely. We have this whimsical, lovely drawing that Monica Zwerger made when she was, when we were working together. If we start at the amino terminal, which is traditionally where we start looking at proteins, there is a Tudor domain, and the Tudor domain is known to bind to H4K20 dimethyl. And then if you continue to follow LBR, there's another region where HP1, the heterochromatin protein 1, binds. So that also binds to chromatin. So there are two parts of the amino terminal part of Lamin B receptor that can bind to chromatin. And that's a very important characteristic of LBR. So the chromodomain of HP1 binds to H3K9 di and trimethyl, and the chromoshadow domain binds to a portion of LBR that has this sequence in the LBR. In addition, there is this transmembrane region, which is not clear how many membrane domains. There's some crystallographic data that indicates there may be as many as 10 transmembrane domains. But the thing that is absolutely clear is that this part of the protein has an enzymatic property, which is a sterol reductase, and it's central in the synthesis of cholesterol. What this is doing in the nuclear envelope, we're not really sure, but it's a very important part of the molecule. There are several medical characteristics of mutations in Lamin B receptor. One is Pelger-Hewitt anomaly, and the other is Greenberg's dysplasia. I think in the interest of time, I'm not going to go into those two things. The one thing that we have, there is a protein, TM7SF2, which is a somewhat shorter protein. It has a homology to this region of LBR, the transmembrane domain, it has serol reductase activity, but it's missing the part that binds to chromatin. So it's very interesting that in the LBR knockdown, our transcriptome data indicates that TM7SF2 increases and presumably is backing up the cholesterol biosynthesis part of the needs of the cell. Now I'm going to show you a tomogram to look at the structure. When we first saw ELKs, and when Howard Davis discovered them in 1974, we were very excited that this would be a structure that would tell us what way, how are chromatin fibers organized, because we're still struggling with higher order structures of chromatin. So we did a tomogram of ELKs. Some of the micrographs for this study were taken in Colorado by Mary Mayhew. The calculations and analysis were done by Mikhail Eltsov. The interesting thing, although we thought we were seeing 30 nanometer fibers lined up in the ELKs, when we did the tomogram, that did not seem to be the case. And we saw what looked like fibers. We traced them here with dotted lines. This is a 90-degree flip of the tomogram. So we're looking in the Z direction and the X direction. So how is the chromatin organized against the nuclear envelope? There are two internuclear envelope membranes. And it looks like a crisscross pattern. We've traced it here with dotted lines. Well, we've got it here with isosurface rendering in panel D. Panel B, C, and D are all the same data. The Z, or the 90-degree flip of this tomogram that's in the white box here. When we look at this in more detail, we found that there are two layers of fibers in the ELKs, probably about 17 nanometers each. But as you see, they're very irregular fibers, so I don't want to say too much about their dimension or the organization of nucleosomes in these fibers. But it looks like there are really two fibers, and that they're maybe organized in some pattern by the nuclear envelope, sort of a hashtag kind of pattern. We figured that cryo-electron microscopy would be a really good way to look at the structure of ELKs, and worked with the people in Wolfgang Baumeister's lab, Julia Mahmoud and Marko Dombrovsky and Peng Shu. They looked and did isosurface rendering of ELKs. And here, very beautifully, you can see the inner membranes, inner nuclear membranes in red, and the outer nuclear membranes in blue. And then the chromatin, of course, in gold. When they measured the distance from the inner nuclear membrane to the brightest region of the chromatin, they got this distribution, and it indicates that the midpoint is approximately 30 nanometers from the inner nuclear membrane. If they measured the distance between the two inner nuclear membrane, they got a value of 58.6, approximately 60 nanometers. So the first thing to notice here is that the distances measured in cryo-electron microscopy where we believe that the preservation of the in vivo structure is much better than in the chemical fixation and embedding of classical electron microscopy specimens. The structure was actually 50% larger than we thought it was going to be. I'm not going to discuss this. It looks a lot like the structure that we saw in the fixed chromatin, looking at 90 degrees here on the bottom of the panel. But although there is some distortion due to the missing data in the Z direction, I think there is some indication that there are fibers running parallel to the surface of the internuclear membrane. And just to emphasize the difference in the structure of embedded, chemically fixed preparations and the cryo-EM, I'm showing here that the distance between internuclear membranes is 34 nanometers for the fixed, and for the cryo-EM, it's twice that distance. So fixation in plastic really does shrink the structure that we visualize in the electron microscope. Now, I can't resist showing you two more micrographs of the surface of chromatin. The panel on the left was taken by Travis Gould at Bates College with his STED microscope. This is a high-resolution light microscope. And the only stain that's on here is DAPI, which is a stain that's specific for DNA. And it is quite remarkable how sharp and how distinguished the surface is compared to the rest of the nucleus. Years earlier, we took an electron micrograph with a DNA-specific stain named osmium-amine. And with that stain, we see a very similar top layer on the surface of the nucleus. So the epichromatin, we think, really has a very different structure from the rest of the chromatin. Let me show you a similar study of cryo-electron tomography. This again was done by Julia Marco in Baumeister lab. If you look at the structure of the chromatin, now this is a lobule, this is not the ELX structure. You have an outer nuclear membrane, an inner nuclear membrane. Here's a nuclear pore. But look at the chromatin on the edge of this. And when they did a density profile, they found that the chromatin had a very sharp peak in the layer that's closest to the nuclear envelope. And it centers at 26 nanometers. There's another peak further in, I would guess at maybe twice that distance. But the structure very soon gets disorganized. And this is the region that everybody's now talking about, liquid droplets and very dynamic kind of chromatin structures. And I think we do have that kind of a structure, but for some reason, the chromatin that is close to the nuclear envelope is more organized. Just quickly, some views of chicken erythrocytes where chromatin is much more highly organized. I think everybody agrees that chicken erythrocyte nuclei, where there's almost no transcription going on. Here are the micrographs of Howard Davis, which show what looks like fibers, cross-section, and along the surface of the nuclear envelope. And this organization seems to extend into the nucleus for quite a distance. In addition, if you do thin sections, grazing sections parallel to the nuclear envelope, you see there's an amazing organization of chromatin. I want to do one more thing before I hand over to Don. I'm sorry, I'm really running over, but I want to mention a method that we've devised for doing some biochemistry on the higher order structure of chromatin. And this is a fun collaboration that we've had with Vladimir Taif. And indeed, this paper just came out this week, so it's fun to celebrate that. If you take live cells and fix with formaldehyde, permeabilize the cells, then sonicate, you can do a normal chip-seq experiment. X here means cross-link, and we only cross-link once, and it breaks up the chromatin into nucleosomes. Antibodies bind to whatever epitope. In this case, it was H1. And so then we can isolate all the sequences that have bound H1 to them. If we fix, permeabilize, and then react with antibody, then those regions that have H1s that are hidden, where the epitope is not available to the antibody, are not going to react. We then find that if we fix again and then sonicate, we can differentiate between those nucleosomes that had those DNA sequences that did interact with H1, but were hidden and were not pulled down by the antibody. So if we now look, for instance, on the sequence of the 7th chromosome, the top 2 rows are X chip, a single fixation. This shows all the regions that interact with the H1.2 or H1.5. We get the gray profile and the gold profile. If we look at the sequences that were pulled down with the XX chip, the double fixation, for H1.2, the blue, for H1.5, the green, we find that there are regions that were highly interactive in the X chip, and therefore had a lot of H1, but they don't show the high interaction with H1 with the double X chip. So these regions demarked by the arrows here are actually hidden H1 binding sites and not accessible when the higher order structure is intact. Briefly, this study, Vladimir did a wonderful job in showing what regions of the chromosome have hidden H1 epitopes. And the amazing thing is that in H1, we've always known that H1 is involved with higher order structure, but this really proves it and shows that enhancers, promoters, CPG islands, look how small the signal is in the XX chip and the bigger signal with the X chip. So the other thing I want to show that here on the end is epichromatin, and indeed, even in epichromatin, the surface chromatin, there are H1 binding sites which are not accessible and hidden by higher order structure. Sorry it took so much extra time, but here is the bridge to the other part of our talk.
Okay. Well, thank you very much Vladimir, thank you very much, Christine, and all your colleagues for giving us a chance to talk. And I only wish that, well, in a sense, you were all here with us now so we could exchange questions and things right away. Okay, so what I'm going to do is continue on where Ada was. She introduced the concept of epichromatin. And what I want to tell about is using antibodies to study epichromatin. The data Ada just showed you was using immunoprecipitation with anti-H1s, but this is going to be a different antibody. It's an anti-nucleosome antibody that was developed by a friend of ours, and let me show you. His name is Mark Monastir. He's at Temple University, and he was working with autoimmune mice that developed antibodies to nucleosomes. And he sent us a whole slew of these antibodies to test. And one jumped out of the group, and it has the name PL2-6. You'll hear that quite a lot in the succeeding slides. So and we found that it appears to stain the epichromatin region specifically. And so these pictures are through the cell cycle of U2OS cells. And you can see that the staining on an interphase nucleus, these are just fixed and permeabilized and immunostained cells, okay? And these are all attached cells. So what you can see is the very fine layer of staining on the interphase cells. And as you progress through the cell cycle and you begin to get the DNA clumping in prophase and late prophase, and the nuclear envelope, we know at this point is beginning to break down, but we're still seeing the staining of some of the surface of the chromosomes. And then we go through metaphase and the reconstruction of daughter nuclei in late telophase, and we can see that the epichromatin surface, the surface of chromatin, persists all the way through the cell cycle. There's an interesting gap here and also seen here, which is where actually the microtubules come in to bind to the kinetochores. Okay, so it's a very interesting antibody. Now, when viewed with confocal and STED imaging in collaboration with Travis Gould at Bates, you can see this is, again, PL2-6 staining, the staining in confocal microscopy and now STED. And you can see that the resolution is really quite a good deal better, as you all know. The resolution in a STED microscope is about 30 to 40 nanometers, and in confocal it's more like 80 to 100 nanometers. So you can see that there's a very thin layer of this antibody staining. On the other hand, when you look at the mitotic chromosome cluster here on the bottom, on the left is confocal, on the right is STED, you can see the average thickness is pretty much the same, but if you look carefully at the surface, you can see it's not as smooth. It's got bumps in it, and there's obviously more surface here. I like to analogize it to the coast of Maine, which has so many inlets that it's a much larger area. And in fact, the antibody stains mitotic chromosome epichromatin much more brightly than the interphase. Okay, so now an immunoelectron microscopy to show you, and this is beautiful work that was done by a very good collaborator of ours who's unfortunately no longer alive, Hans-Walter Zenkraft, who did beautiful immunoelectron microscopy. And these are ELKs at a low mag, same ELKs at a higher mag. And these little tiny dots here are little gold particles and show you that the ELKs stain with PL2-6. Likewise, here's a region here, low mag. You can see the nucleolus here, and there's a thin layer of chromatin right between that and the nuclear envelope. And that thin layer of chromatin is also staining with the PL2-6. Okay. Now, just the top row is what I want you to focus your attention on now. And this is a repeat of what I've shown you, an interphase nucleus staining with the bivalent PL2-6. It's a bivalent antibody. Now we used an additional antibody, anti-H1.5, in the same experiment. It's a bivalent antibody, but it stains throughout the nucleus in a punctate manner. Is DAPI staining, and then the merge, and you can see that just the merge of the previous pictures, a little bit yellow here, suggesting that there is some H1.5 also binding in the epichromatin region. Now, what we did was we made FAB, or monovalent fragments of the PL2-6 antibody. And when we stained that on interphase nuclei, it's punctate, just like the bivalent H1.5. And when you look at a merge, you can see there's some overlap, but there are a lot of dots that don't overlap. Now, when you look at a mitotic chromosome cluster, so here you see the clustered mitotic chromosomes, and they all have little tiny dots on them. And this is the FAB staining. With the H1.5 bivalent antibody, we're still seeing dots. And even with the DAPI, you can't quite see it, but you can see dots there. When you look at, these are serial sections of this one cluster of mitotic chromosomes, you can see that some of the dots, I know they're tiny and it's probably impossible for you to see, but some of the dots are red, some of the dots are green. So there isn't a complete, there isn't, not every particle has H1.5. In fact, there are 3 different H1s in these cells. Now, we want to call these dots chromomeres. It's a name that goes way back to the end of the 19th century, and they were first seen by Walter Fleming in his, this is before people were taking digital images in their microscopes. And this is a mitosis, and it shows these wonderful dots along mitotic chromosomes. Walter Fleming coined the terms chromatin and mitosis. He did not actually use the term chromomere. That came later, a little bit like, I guess, new bodies and nucleosomes. Okay, so now what I want to do is show a tangential section, just a grazing section like what Ada showed you earlier of U2OS cells. This is imaged in a 3D SIM microscope, which doesn't have quite the resolution of a STED microscope. So here you have a midsection. You can see nice thin staining of the epichromatin and DAPI blue staining in the middle. This is a low mag of the tangential view. And then this is a much higher mag of the tangential view. And if you look very carefully, and I hope you can see it, you can see there's some thin lines here, parallel to one another, very reminiscent of what Howard Davis saw in his tangential sections of the chicken erythrocyte nucleus. And we've estimated the spacing here between the center to center spacing between these fibers is approximately 160 nanometers. So I'm going to ask you to remember that wonderful number because it's going to appear later on in my part of the talk. Okay, so now we want to find out what, in fact, is PL2-6 actually binding to. And in order for you to understand that, I have to show you this beautiful view of the nucleosome following crystallography. And I do want to mention that this was also much earlier done in Oak Ridge. And what we have here is that the H2A and H2B constitute an acidic patch. The acidic patch is filled with glutamics and aspartics, and it is in fact the place where the the PL2-6 antibody binds, very much like other proteins that are known, the H4 N-terminal tail, LANA, SENPC, which is part of the kinetic core, and HMGN2 and others. So let me move on. This is the consequence of a very productive collaboration with Lamin B at NIH. He took the known peptide sequence of the variable region of the heavy chain in PL2-6 and the light chain in PL2-6, made a single peptide antibody and reacted it with recombinant mononucleosomes. And this is the analysis. And let me go very quickly through this. This is the critical arginine that's seen in the proteins that bind to the acidic patch. It's arginine 124 of the heavy chain. It's the heavy chain variable region, third variable region. And it is in fact binding to a glutamic acid here on H2A, that's E61. It's also binding to an aspartic D90. That's no,
Pink is H2B.
Oh, this is H2A. Did
Yeah,
I speak?
yeah.
Oh, okay. This is why we work together. And you can see there's also an interaction with glutamic also on H2A. Okay, so those three sites are on H2A, but there are other sites on H2B that are interacting with. So we now know for sure that this antibody binds to the acidic patch. Well, how do we get the difference between the monovalent antibody binding in chromomeres and the bivalent antibody binding in the epichromatin region? Well, we just really have speculations. We have not been able to yet do a crystallographic or a cryotomographic study. Anyway, so the idea is that we have here, here's my cursor. The idea is that the bivalent antibody also has a dyad axis in it, and the epitope binding regions are rotated 180 degrees from one another by the dyad axis. And if you look at the zigzag arrangement of nucleosomes, which is one possible arrangement, then you would find that the acetic patch polarities are 180 degrees rotated from one another. And we sort of said, well, wouldn't it be nice if this polarities, complementarities matched and you could get good binding? So the next slide is hypothetical, but very good modeling done by a crystallographer we worked with in Munich. And this shows you, move this around, and you can see this is the PL2-6 antibody, an FAB fragment, the FC fragments going in the back. This is a dinucleosome. These red spots are the location of the acidic patch. And here coming around the bend, we see the FAB fragments interacting with the acidic patches and the FC fragment coming around. Now, when you cut with papain, which is what you do, you cut right in here and you get a single monovalent or a single monovalent and you get the FC. Okay, moving quite along. Oh, I don't want to do that again. How do I hit again? Oh, yeah. All right, now to change a fair bit, but really we've talked about it. This is an example of using the XX-ChIP-seq method that Ada introduced for you. In fact, this was the first time we developed this method for this purpose, to study the PL2-6 antibody. So what these tracks are the enrichment or depletion of what turns out to be, well, what did I lost my hand? Okay, turns out to be what we'll call epichromatin regions. This is the enriched epichromatin regions. This is chromosome 1, the Q arm and the P arm over here. And we have duplicates of undifferentiated cells, duplicates of the granulocytic cells. We have a single example of the macrophage. And then we have two examples from another antibody, which is available commercially, 1H6, which apparently does the same kind of binding, but no one actually knows exactly what it binds to. And so what you see here is that as we go along the first chromosome, there's a whole stretch here which is highly enriched in epichromatin. These are the regions that are brought down by the antibody. And these regions are not brought down by the PL2-6. And likewise, and here's another stretch here, which is highly enriched in epichromatin. Now, if you align these epichromatin-rich or epichromatin-poor regions next to the tracks of the retrotransposon ALU, you can see that the epichromatin is highly enriched in ALU and is not enriched in L1, another retrotransposon. Likewise here, highly enriched in ALU and somewhat enriched in ALU, but not in the regions where L1 is. Okay, so let's move on. Let me summarize what still is our situation, how we think of epichromatin. In HL60S4 cells, it turns out that the total amount of genome that is represented in epichromatin is only about 4% of the genome. There's a clear discontinuous distribution on the chromosomes. Let me see where I'm going to try and, Vladimir, can I say that if you want me to end, can you send a signal to us somehow? Anyway, okay, so we've got discontinuous distribution on chromosomes. There seem to be about 6,500 positions that are common during differentiation. So as I showed you, the epichromatin pattern looks very similar in the differentiated cells. There's a tenfold enrichment of transposon ALU. There's an enrichment of stable CpG methylation, and there's a paucity of repressive and permissive histone modifications. Okay, so now I want to show you how does this relate to high C, which you guys now all know about. It's become a very well-known method. So again, this is chromosome 1 here. This is the Q arm, and this would be the P arm here. I just want to show you, here's a region that's highly enriched in epichromatin. And if you look here over, you can see that there's either a TAD or a sub-TAD, I don't know quite what you call it. So there's some correspondence between epichromatin regions and these regions of high internal cross-linking. So that suggests that this TAD or sub-TAD has parts of it that are represented in the epichromatin region at the surface of these cells. And the other thing you'll see, as has been known for a while, is when cells differentiate and they change their transcriptome pattern, there's very little change in the high C matrix maps. Okay, so let me summarize this part. Epichromatin, the surface of chromatin, exposes nucleosome acetic patches recognized by the bivalent antibody. The interphase epichromatin represents only about 4% of the genome. Epichromatin has a paucity of promoters, transcription start sites, exons, permissive and repressive histone post-translational modifications. It's enriched with introns and intergenic regions. It's about 80 nanometers thick. It's smoother in interphase cells than in mitotic epichromatin. And chromomeres expose the acidic patches throughout the nucleus and bind monovalent PL2-6. And lastly, on this point, that some TADs reside in epichromatin. Okay, so I'm coming to the actually very last part of the talk. There's no way that I can interact with them and find out if I've completely lost the audience.
You have not lost the audience. We were just
Oh,
letting you finish. Sorry.
okay. Oh, I don't know. You know, I don't know. I mean, you know, actually, we're surrounded by fog, and we're going to have thunderstorms in a little while. So,
Yeah.
I'm not kidding.
We're still
All
here
right.
and you have some questions.
Okay. Are there questions?
There are, yes.
Okay.
But we,
Do you want to keep going for a few minutes?
Can I ask the person to hold on to the question? I promise we'll answer every question we can, but
Absolutely.
I want to get to this part, which we are very excited about. And I've lost my cursor. Oh, here I am. Okay. We wanted to find out what happens when you treat these cells with hyperosmotic stress. Now, when you take mammalian cells and you expose them to either sucrose or sodium chloride, those do not get into the live cells. Rather, water is drawn out of the cell by the high osmotic pressure. The cells are dehydrated. Now, I think you'll see that if you can hold on to the end of what I'm going to say, that it actually turns out to be really interesting. But what we see in the microscope, this is now immunostaining with PL2-6 and DAPI. On the left is just cells growing in medium. This is HL60 cells. You can see mitotic chromosomes here that have a bright bright epichromatin staining. You can see lots of interphase nuclei. This is low mag. I'll show you high mag in a minute. You can see hazy interphase chromatin surrounded by the epichromatin. Now, in 300 millimolar sucrose, that's twice isoosmotic. There are phase changes taking place galore in these cells. The mitotic chromosomes turn into these little tiny jelly beans, very highly dense in DAPI. I'm going to show you a high mag picture to prove this to you. Okay, yeah, I'll go to that right away. Okay, so at high mag, this is in the absence of sucrose. You can see the mitotic chromosomes surrounded by epichromatin, and you can see the substructure of mitotic chromosomes. If you go to 300 millimolar sucrose, you get this, we call it congelation, because it almost looks like a ball of gel surrounded by the surface epichromatin. The interphase chromatin goes from hazy to clumpy, or we also use the word congealed or congelation. Now, how do we know that we're actually seeing this as a, how do we know this is a, did I lose my cursor? Oh, here it is, okay. How do we know this is a mitotic chromosome? Okay, so what we did is we used another antibody, H3S10P, which is a very well-established mitotic marker antibody. And here you can see in low sucrose, no sucrose, you can see nice mitotic chromosomes stained with this mitotic marker. None of the interphase chromosomes are stained. And you can see here the mitotic chromosomes in 300 millimolar sucrose are stained with, at high mag, with the marker from mitotic chromosomes. Okay, so this is in no sucrose, H3S10P, and this is in high sucrose, H3S10P. Okay. Okay, now now I wanted to show you that we're getting really some interesting phase transformations taking place. Ki67 is a well-known marker also for mitosis. It has been shown in beautiful studies. Names will escape me, but as usual now. But you can see that Ki67 is on the inside of a cluster of mitotic chromosomes. It's the function, it serves to separate the mitotic chromosomes. And in this, let's see, I want to get the name. Yeah.
Daniel.
Oh yeah, Daniel Gerlich and Jan Ellenberg did a beautiful study on this. They they show that this protein acts as a surfactant separating the mitotic chromosome arms. And the thing is that what happens in high sucrose is there seems to be phase separation and demixing, and in the mitotic chromosomes coalesce, and the Ki67 goes outside the epichromatin region, and it also can be seen in the interphase cells, but I'll skip that. Now, it isn't only sucrose that does this effect. This shows you that 150 millimolar sodium chloride, which is approximately isotonic with the osmolarity of sucrose, is having an effect. Now we're getting close to the 160 nanometer number that I wanted to tell you about. So here we have, essentially we're looking at DAPI staining. Excuse me. We're looking at DAPI staining. These are control cells in no sucrose, just tissue culture medium. And you can see that with DAPI staining in a STED microscope, you can see this very thin rim of epichromatin. You don't really see it in the mitotic chromosomes. When you go into 300 millimolar sucrose or 150 millimolar sodium chloride, and you get the congelation taking place, the main thing I want to show you is if you do a radial distribution analysis, which is an analysis of the distance between neighboring dense, intense regions. So that would be DAPI staining. How close are the closest neighbors and how prevalent are they? What you see is you see a peak in all of the cases, regardless of whether the nuclei have been condensed with or congelated with sucrose or salt. The position is just about 160 nanometers. Yes, that work was done, thanks. That work was done in collaboration with Travis Gould. So here you have the data, and they average out to about 160 nanometers peak density. And this is called density-density pair correlation. Now we're getting to what I think is some of the most interesting stuff, is RAD21. RAD21 is a component of cohesin. And you know cohesin is very important for developing these loop domains that are observed in high C. So in the absence of sucrose, you can stain for RAD21 throughout the interphase nucleus, and often you can see it in the mitotic chromosomes. When you go to 300 millimolar sucrose, the world changes. RAD21 now seems to be segregated outside of the mitotic chromosomes here, and has segregated into the region just about where epichromatin is. And this is two examples.
But
now, pardon me?
But in clumps.
Yeah, in clumps, right. The point is we're getting clumps of RAD21. Now, what's the significance of this? The significance has been shown in earlier studies coming out of Erez Lieberman Aiden's lab, and most recently some work that Marion Kremer and Thomas Kremer have been doing with Erez Lieberman Aiden and lots of other folks. And that is when they use this auxin method for degrading RAD21, they lose the loop structure, the loop anchors, and this method called aggregate peak analysis, what you do, you pile up like 6,000 loops, and you can see that there is this spot. I didn't get a chance to point it out when I showed the high C, but it's at the apex of these loops in high C, many of them, not all of them. You see this fine peak, and now when you degrade cohesin by this method with degradation that they previously used, then you see a gradual disappearance of these anchor points. Well, this is now with sucrose, we're seeing it. So we're not degrading the RAD21. Rather, it seems to be coming off and segregating, demixing and segregating. Well, you're wonderfully patient and you haven't said anything. A couple of more things. What I want to show you is that we collaborated with Marion Kremer and Irina Solovay, and they did some beautiful chromosome paints. And this shows that the chromosome territories actually are retained during this procedure. So the chromosome territories are maintained and do not appear to intermingle when you compare 0 millimolar sucrose and 300 millimolar sucrose. So this is an interphase nucleus and then a congealed interphase nucleus, a mitotic cluster and a congealed mitotic cluster. But you can clearly recognize the chromosome territories. They haven't become a ball of mixed colors, so they don't intermingle. So let me summarize hyperosmotic stress. Hyperosmotic stress dehydrates live cells. The cells shrink to about 2/3 the initial volume. Cell solute concentrates about 50%. The cells become less deformable. We demonstrated that just a few months ago with Joachim Guck's group in Erlangen. Yes. Dehydration leads to phase separation, and the interphase chromatin and mitotic chromosomes congeal. We get proteins dissociating and demixing, but epichromatin remains at the surface of the congealed chromatin. And the interesting thing, and I promise you this is the absolute last thing I'm going to describe, the cells are still alive and they exhibit gene expression. And when we looked at our transcriptome data coming back from cells treated with sucrose, we could not believe what we saw. This is the transcriptome data of the histone genes. And it looks like the histone genes all increase their transcript level in sucrose. And the thing that really shocked us is that they shouldn't have, because we had purified the poly-A mRNAs. And what we're really looking at is the replication-dependent histone transcripts, which have stem loops. And we didn't purify stem loops. So we're seeing suddenly something happening to the histone messages. So we quickly contacted Bill Marsloff at UNC, who's a friend of ours and who has built a beautiful career looking at histone messages, and said, what's going on here? So he said, oh, that's been seen before in a few cases. And in fact, they had seen it where you play with some of the proteins that are involved in modification of the stem loop. And if you get defects or knockouts, then you begin to see sort of a default situation where these replication-dependent histone messages are turned, get a poly-A on them. Okay, and so at the same time, you can see that the replacement histones, which do not have stem loops on them, are in fact all decreasing. I'm not going to go through this. So how can this happen? Well, when we looked at our gene ontology analysis of what happens to the transcriptomes in high sucrose, we found that the category that was the most depressed was the nuclear bodies category. Nuclear bodies, that's like Cajal bodies, PML bodies, the nucleolus, paraspeckles, but it also includes what's known as the histone locus body. And so we haven't really studied this in the microscope, but we looked at the components of the histone locus body. So it looks like this is SLBP, which is one of the many proteins. These are all transcripts now of the histone locus body. it looks like the transcript level is decreasing, and this is an essential protein. It's the RNA at this point. This is an essential protein for protecting the stem loop during S phase and during the translation of the histone message. So we are supposing that the histone locus body is also undergoing some kind of phase separation, and it's losing its capability to protect the stem loop, and we're getting some kind of a default mechanism taking place. So this looks like, and it should be next to, this is a summary talk. So a summary slide. So what is stable through all in these cells, nuclei and cells? The stability that we've seen is the differentiation transcription program, the TADs during differentiation, the epichromatin composition seems to be stable. The ELK structure, even though it's a flexible structure, seems to be quite stable. The chromosome territory seems to be quite stable in the sucrose effect. Whereas what's plastic? Well, the cells are highly deformable, especially the RNA, the RA-treated cells. The nuclei and the cellular deformability is noticed during migration, and the transcriptome changes during migration. And this is a paper that we published with a group from New Zealand. The transcriptome really changes as these cells crawl through narrow pores, and it also changes during hyperosmotic stress. And the ultimate, okay, this is to tell you what we would like to work on if we can. In the future, we want to find out what's the basis of the 160 nanometer distance. We want to find out if the epichromatin composition is a lineage-specific signature for each type of cell differentiation. We want to find out if the hyperosmotic sucrose effect is reversible. And we want to see what's the basis for this change in the replication-dependent histone transcripts. Finally, this is the slide where everyone shows who's in their lab. Well, I think you can see, you've seen it. I mean, it's just the two of us, but these are just some of the wonderful people that we've done collaborations with over the many years. And we hope to keep doing collaborations. And I really appreciate you guys listening to the end of this. And that is the last slide. How do I get out of it?
I don't know, somebody else should help us get out of it.
Oh, yeah. Is there anyone still there?
You can keep this slide. Thank you very much. Thank you very much. We'll have the Q&A now.
Yes.
Okay, sorry, guys.
Thank you so much. That was such an inspiring talk to hear both of you. And I loved how when Ada chimed in and everything during Dawn's talk, that was really fun. And the images are just beautiful. So it's really exciting to see all of your work. We have quite a few questions. I want to remind everyone that you can, in fact, raise your hand and ask a question yourself. So if you would like to do that, please do. And just to start, I will go ahead and ask a question from Jeremy De Silva. So he asked, and I believe this was during Ada's portion of the talk, are the different lobules only connected by ELCs as it appears in the electron micrographs, or is it an artifact of taking a Z-plane? In other words, are they connected by more nucleus in a direction projecting out of the plane?
Is my microphone on?
Yes.
Okay. It's all one nucleus, so everything is connected. Sometimes the ELKs sort of form a bubble on top of a
lobule?
Of a lobule,
Yeah.
thank you. everything is connected, and you see all kinds of connections. Sometimes there's ELKs actually connect to lobules. Sometimes they're just on top of the lobule. It's all one nucleus.
Can I, if you don't mind, you're going to get two of us. These are sheets. These should be thought of as sheets like a sandwich, a big sandwich of sheets with internuclear membrane on both sides and the chromatin in the middle. And there's then the outer nuclear membrane, of course, outside of it. And the thing is that we think that Lamin B receptor somehow regulates the growth of these internuclear membranes. We have no idea how. And it just grows. It's like cancer of the internuclear membrane. It just grows like crazy and keeps spreading the chromatin attached to it.
Thanks.
Retinoic.
Oh, sorry.
I was going to say that retinoic acid has been used to treat people with cancer, and that also induces ELKs and lobules, and then the cells die, which is the natural pathway for granulocytes. They go into apoptosis. So these have been seen in natural circumstances besides the tissue culture cells.
Cool. I will ask a combined question from Igor Prudovsky. He also says
Hello,
hi.
Igor.
Hello, Igor. You know where Igor is.
He's
And
in
Igor,
there.
if you want to raise your hand, say hi, go ahead and raise your hand and we can unmute you as well. But he and, oh, hi. He and Hilmar asked a similar question where, let me find it, sorry. Okay, here we go. They want to know if there's any selectivity of chromosome presentation in ELCs, and similarly, what kind of chromatin are in ELCs, and whether a technique like ORCA, which we heard about last week, the optical reconstruction of chromatin architecture, could be helpful in figuring this out.
Yes, very nice. That would be fun. I'd love to see that.
We don't know. We don't know.
From the first day we've seen ELKs, people have asked us what the sequences are in the ELKs, but we don't really know.
But I think now a method is available where we could do it. I mean, the problem, we couldn't figure out how to do the experiment because you'd have to isolate the ELKs in order to determine what DNA is present in them. And it just never seemed easy enough to do. But I think maybe it is a good time. And hello, Igor.
Hello. I have additional question. Is there any transcriptional activity in ELKs?
I don't think we've ever seen any indication of transcriptional activity, and all the chromatin that we ever see in ELKs is pretty well condensed, so that is in agreement with our lack of observation.
Yeah. You know, there is, I don't know, right.
We've lost our image.
I don't know.
I'm gonna unmute Pallavi Mustafi.
Sure.
Oh.
We've lost
Hello.
images of people.
Yeah. Can you hear me?
We can hear you, but we can't see anybody.
So it's kind of okay.
Go for it.
So I was just continuation to about ELKs only. I was wondering, have you observed the fiber-like structure changing across like during the course of differentiation, or is it only the lobulation that is changing and not the structure of ELKs as such during differentiation? And in the lamin knockdown cells, do you see any change in the ELK formation?
We only see the ELKs when we differentiate the cells to granulocytes with retinoic acid. We don't see it when we differentiate to macrophage. And there really is no connection between the two differentiation pathways.
You pointed out we do see them with vitamin D3 with the monocytes.
Right. There's
Yeah.
another. These HL60 cells can also be differentiated with vitamin D3, and then they become monocytes. The monocytes also have ELKs, but many fewer than the granulocytes.
And the structure is similar, or is it different?
It's whenever we see ELKs, their structure is very much the same. Of course, I mean, we see different kinds of nuclear structures everywhere, but then they're not a sandwich sheet between, chromatin sheets like that. We also, what is the situation when we get multiple ELKs?
Oh, that's with BL2,
yeah.
with, yeah.
Yeah, we've lost our talk. I have a slide I could show you of multiple layers of ELKs, which we see in BCL2 overexpressors. And that's a really amazing structure. So if it's many layers of ELKs piled on top of each other.
Thank you.
Just
Cool.
There is another case. Yeah,
wait, let me.
There's another case where ELKs have been described, and that was Brian Burke, who described in mitosis. I think it's the beginning of nuclear envelope reformation after mitosis, that structures like this have been seen. And it may be due to its Lamin B receptor is one of the first proteins to adhere to decondensing mitotic chromosomes. And it may be that this is tied up with, in mitosis, the reestablishment of the interphase nuclear envelope. And yeah, so Ada, you wanted to...
I just thought I'd show you, we have in our spare slides a study we did many years ago with BCL2 overexpressors, and we get these incredible structures.
Can you see these? Can you see this?
Yes.
Okay. Christine, this cell line came from Fred Hutchinson lab.
Oh, that's great.
I was going to ask you if you could find it in the freezer there. We've lost it.
I could ask.
But you see, can you see my arrow? It's pointing to an outer nuclear membrane. Then there is a layer in a nuclear membrane, chromatin in a nuclear membrane, another in a nuclear membrane, chromatin in a nuclear membrane, and a third layer like that. So in these cells that can grow for much, much longer, don't go into apoptosis as quickly, we see these incredible structures.
Mm-hmm. Mm-hmm.
I can't explain what's going on here, but
Thank you.
Cool.
Sure, glad to.
We have quite a few questions. I'd like to get...
We're with you as long as you want.
Oh, thank you. So the next question I'm going to ask on behalf of Ulduz Afshar, and they asked, can this XX chip be used for other nucleus-associated proteins such as transcription factors or histone marks? And have you used any other factors outside of H1 and PL26?
The answer to your last question is we have not. The answer to your first question is we don't know. The answer to your middle question is we hope you do it, because we would like to see this method become useful. The idea is to find out what epitopes are hidden if you can preserve the higher order structure that is the first fixation preserves higher order structure, and the second fixation preserves the antibody bound to regions, because then you have to sonicate it and fractionate it. So no, we haven't gone further, unfortunately. We'd like to do
But what
more.
we'd love to see this applied to
Yeah,
other
yeah.
proteins, because
Yep.
many proteins are quite mobile. So if you don't do the second fixation, they'll bind after the fact.
Yeah, it would be really cool to see. So the next question is from Srihari Nikki, and they ask, "Hi, I really enjoy the talk, and I was wondering if there are images involving protein-specific tags, like using a halo tag that's used for super resolution microscopy. Could this be done with epichromatin? Would that improve the resolution after, like, as opposed to using antibodies? And is there a way that one could tag the acidic patch for this type of imaging in live cells?"
Yeah, it would be nice. One of the problems is that it's not easy to transfect HL60 cells. But since we've seen epichromatin in all kinds of cells,
Everything.
including plant cells,
But, right.
there's no reason why that isn't doable.
We could even see it in yeast protoplasts, if I
What?
remember.
Really? Oh, cool.
I mean, look, the epitope is just the acidic patch.
Oh, right. Yes, and that's highly
And histones have been conserved so highly in evolution. Right. No, we haven't tried any of those methods. Sorry. We could.
And then the next question is from Rebecca Ellizer. And she asks, have you tried pulling down with PL2-6 followed by mass spec to see if there is an enrichment of histone variants in epichromatin?
We obviously have not done that.
We have done that. We've done one of the problems we have is to convince someone who's got mass spectroscopy to work with us. But we have done it, I mean, they're such busy people. Everyone is knocking on their door trying to get the analysis. But we did work with an excellent mass spectroscopist in Innsbruck, Bettina Sarg, who looked at these were the H1s, right? I can't remember. Yeah, no, we did. What we did was we were doing cross-linking of various chromatin-associated proteins, and we would run them on gels and cut them out of gels, and that's actually in the paper on sucrose. It's that we just published recently in Nucleus. So no. Unfortunately, a lot of the things that we would like to do,
yeah, that would be
still need to be done.
Need to be done, and that's why you guys are in the field.
It's great that both of your talks have sparked so many cool questions. We do have another one from Rasim Bardku. Sorry if I messed up your last name pronunciation, or both names. If you would like to raise your hand, actually, Rasim, and ask your question yourself, go ahead and do that if you would. Yes, perfect. You did that. Here we go. Oh.
Hi, can you hear me?
Yeah.
Yes, but we can't see you. All right.
Okay. So my question is regarding this epichromatin. There's this whole nomenclature starting from 1974, Coffey and Berezny, Sheldon Penman, Jeff Nickerson, based on the nuclear matrix, the insoluble fraction of the chromatin.
Yeah.
I would like to ask you your general opinion about the concept of nuclear matrix and how it relates to the epichromatin. And the other question is the epichromatin you described, is it RNA sensitive?
Mm-hmm.
Well, you
Well, okay, let's start with the second question. We haven't done it. Okay.
The RNAs.
The RNAs. The first question, what's the relationship to nuclear matrix? Well, we've lived through all of that time, and we've witnessed it, but we haven't really done much in that. The nuclear matrix is is, well, first of all, epichromatin is chromatin. So it's, and all concepts of the nuclear matrix were really of proteins that were were not really, well, they might be
bound to.
Yeah, they would bind to chromatin. Honestly, we haven't pursued nuclear matrix. It requires so many strenuous treatments of nuclei. So I'm afraid, again, we can't really satisfactorily answer your question. I think there's some reason to be skeptical about the concept of a nuclear matrix. And it's been sort of in the dark corners for a long time. No one's really brought it back out. So I can't really answer your question. Sorry.
But
so regarding the RNAs, I'm sorry.
Yeah, go ahead.
Yeah, I did want to say about the RNAs, although we've never done this experiment. For many years, doing the electron microscopy, I always felt that sharp layer under the nuclear envelope that today we're calling epichromatin always looked a lot like RNA to me in the electron microscope, but I've never been able to really show that it is RNA sensitive.
Mm-hmm. Mm-hmm.
And now that we have the cryo-EM that Yawen Bai has done to show that it's really the acidic patch that is binding to the antibody. I think I've sort of lost the energy to think about the RNA being there, but it still would be a great experiment to see how the surface changes with RNase treatment.
If I can chime in again, Ada showed you a beautiful picture that we took many years ago of the electron microscope DNA specific stain. It's called the Lamin B. And we worked with a fine electron microscopist in Bologna, Massimo Derenzini, who used it as an RNA specific stain. And in fact, we have used that as an RNA specific stain when we, in our former careers, were looking at the Balbiani ring of this big puffed gene in Chironomus, and using RNase and I think following it with the osmium-amine. So yeah, these are doable experiments now, but we haven't done.
If you send us your email, I'll look through some of the data and see if I have any RNase treatment. How's that?
Okay.
You know, if you want to remain.
I'm happy to do that.
Yeah.
Thank you very much. I appreciate it.
Sure.
Okay, so that was fantastic. If we can just do one last quick question from Ben Weekly. He asks if there are any histone variants already known to be specific to the epichromatin.
Not by us.
Maybe someday someone will find it.
You know, a lot of people have. I think whenever you do an immunostain, you find it throughout the nucleus. You know, it doesn't go just to the rim, which doesn't mean that there might not be an enrichment of one particular modification.
You know, when we did our original stuff with PL2-6, the XX chip, that was ChIP-seq. So we purified the DNA to analyze what DNA is brought down. One, in principle, I guess, could look at what proteins are present on that and not go to the point of the DNA purification. We didn't do that.
Mass spec again.
Yeah, mass spec again, right.
That's fantastic. Thank you so much. I want to before Vlad speaks again, I just want to thank everyone for staying with us. This was really fun to have a chat with Don and Ada and hear their wonderful work and even some history thrown in. So that was really cool. Trainees who are attending the trainee meeting, feel free to go ahead and take like a few minutes break, and I'll start the Zoom meeting for that in a few minutes.